Here we are again with news from the website prostatewarriors.com.
We are.
It is January 2026, and, if you've been following the landscape of prostate cancer research, the start of the year is usually when the floodgates open.
It really is.
There's just a massive amount of data dropping all at once, and honestly, looking at the stack of papers we have today, it feels different this year.
It does feel different. We've been tracking this field for a long time. And, usually an update might be one slightly better hormone pill or maybe a tweak to a dosage.
Right. Incremental changes.
Exactly. But looking at this stack of clinical trial updates, what strikes me isn't just the volume, it's the fundamental shift in strategy. We're not just looking for better blockers.
Look, they're looking at something else.
We're looking at a landscape that's shifting and shifting rapidly towards what I'd call ultra-precision and, frankly, biological subversion.
Biological subversion.
Yeah.
I like that. It sounds like StyCraft. Our mission today is a rapid-fire, deep analysis of these updates for January 2026. We're going to cover the full spectrum, from major phase 3 breakthroughs that are challenging the standard of care right now, all the way to some futuristic nanotechnology that sounds like it was ripped from a sci-fi novel.
But it's actually showing results in the lab. And just to set expectations, we were going to be heavy on clinical trials today. That's where the rubber meets the road.
Okay.
We're seeing treatments moving way beyond standard hormone therapies and into precision radiopharmaceuticals and some truly novel biological mechanisms.
So let's not waste any time. We have to start with the heavyweight fight. There's a new phase 3 trial, has everyone talking, BNT324.
Also known as DB1311.
Yes.
Yes. This is probably the most significant immediate-term development.
What is that?
Because BNT324 is entering a head-to-head phase 3 trial against Docetaxel.
Okay, hold on. I want to pause right there and unpack that phrase. Head-to-head against Docetaxel. Docetaxel is the chemotherapy standard, right? That's the reigning champion.
It is. It's the one you call in when things get tough.
You don't usually see new drugs try to knock it out in round 1.
You really don't. That's a very astute observation. Docetaxel has been the backbone of treatment for metastatic castration-resistant prostate cancer, or mCRPC, for a long, long time.
It's a blunt instrument.
A very blunt instrument. It kills dividing cells, period. Usually a company tests a new drug after chemo or before it or maybe in combination.
Right.
For a new drug to step into the ring and say, I want to challenge the champ directly, that shows a tremendous amount of confidence.
So they think they can win. Now, BNT324 is an antibody-drug conjugate, an ADC.
Right.
We've called this the Trojan horse approach.
And it's still the best analogy. You have an antibody that's like a delivery driver guiding the drug to a specific address on the cell.
And once it's inside,
it releases a chemotherapy payload, the soldiers inside the horse. BNT324 specifically targets a protein called B7H3.
B7H3, why that? I mean, we hear so much about PSMA and prostate cancer. It feels like PSMA gets all the press.
It does, but diversity is survival. Not every cancer expresses PSMA, and B7H3 is often found on cancer cells that are particularly aggressive. But the real excitement here comes from the data they generated in the phase 1/2 program. They were looking at patients who are notoriously difficult to treat.
These were the heavily pretreated mCRPC patients.
Heavily pretreated. We're talking a median of 3 prior lines of therapy.
I just want to make sure we appreciate what 3 prior lines really means. These aren't guys just walking into the clinic. They've been fighting this for a while.
Exactly. They've been through the ringer. Nearly all of them had seen docetaxel already. They'd seen next-gen hormones like enzalutamide.
And even some of the newer stuff.
A meaningful number, nearly half, had even already tried PRP inhibitors or lutetium-177. Usually in a salvage setting like this, you hope for modest results.
You're just hoping to stabilize things.
Right. Maybe by a few extra months, but that is not what they found.
What did they find?
They saw an objective response rate of roughly 42%.
42%.
Yeah.
In a salvage setting.
Yeah.
That sounds incredibly high.
It is exceptional. For context, response rates in this setting are often in the teens. Finding a drug that shrinks tumors in nearly half of these patients is rare.
That's an understatement.
But even more impressive to me was the disease control rate, about 95% of the evaluable patients.
Wait, 95%? That implies almost every single patient saw their disease either stop growing or actually shrink.
That's what the data shows. And remember, that includes patients who had already failed on lutetium-177. The median radiographic progression-free survival was approaching 8 to 9 months.
That's huge in a salvage setting. Getting nearly a year of control. It looks like data you'd see from an early line drug.
It really does.
What about side effects? What's the cost of doing business here?
The profile looks manageable. They saw some hematologic events, issues with blood counts, and some nausea, which is common. But importantly, no treatment-related deaths in the prostate cohort.
So, high efficacy, manageable safety. It's easy to see why it got fast-tracked.
It's a very, very promising start for 2026.
Okay, so BNT324 is definitely the one to watch. Now, let's pivot to another showdown. This one feels a bit like a physics experiment, the ANDROMEDA Phase 2 trial.
Ah, the ANDROMEDA trial, yes. This is a fascinating study because it addresses a very specific window in the disease.
Recurrent oligometastatic prostate cancer. Oligometastatic means just a few spots, right?
Correct. Usually 1 to 5 metastatic lesions. The cancer's back, but it's still contained. The standard approach is often SBRT.
Stereotactic body radiotherapy, basically zapping those visible spots with a radiation beam.
Exactly. But Andromeda is adding something else to the mix.
This is what I'm calling the alpha versus beta matchup.
A good name for it. They are combining that SBRT zapping with radioligand therapy, and they're comparing 2 different radioactive isotopes. On one side, you have the beta emitter, lutetium-177, specifically Pluvicto,
And on the other.
the alpha emitter, actinium-225.
So for those of us who maybe slept through advanced physics, what's the real difference between an alpha particle and a beta particle when it comes to killing cancer?
Think of it in terms of range and impact power. Beta particles from lutetium are small electrons. They travel several millimeters through tissue.
So there's a spray, what you've called the crossfire effect.
Right. You hit the tumor cell, and the radiation sprays out to hit the cells next to it. It's like a shotgun blast.
Okay, lutetium is the shotgun. Got it.
But that spread can mean off-target risks. Now, alpha particles, like actinium, they're massive by comparison. They travel sub-millimeter distances.
So much shorter range.
Just a few cell diameters. But they have extremely high linear energy transfer.
Shorter range, but a much heavier punch, like hitting it with a wrecking ball instead of a shotgun.
That is a great visual. A wrecking ball. Extreme potency is the key. An alpha particle hit causes complex double-strand DNA breaks that are very, very hard for a cell to repair.
So the hypothesis for Andromeda is that the alpha wrecking ball is better than the beta shotgun.
The hypothesis is that a single cycle of the alpha therapy might outperform 2 cycles of the beta therapy. But here's a nuance. SBRT hits the visible spots. The radiopharmaceutical is there to hunt down the invisible seeds.
The micrometastases, the stuff you can't see on the scan yet.
Precisely. It's a cleanup operation. And if the alpha emitter is more potent at short range, it might be better at killing those tiny clusters.
And the ultimate goal is to delay the need for androgen deprivation therapy, ADT.
Absolutely. Delaying ADT is the holy grail for quality of life.
Nobody wants those side effects if they can avoid them.
And if this alpha strike can clear the board, we might buy patients years of drug-free survival before they need to start hormone therapy.
Okay, so that's alpha versus beta. Sticking with radiopharmaceuticals, there's another update. SKL35501.
Yes, an investigational new drug application, an IND, that was just cleared.
Now, usually when we talk radiopharma, it's always PSMA this, PSMA that. But SKL35501 is doing something different, isn't it?
It is, and this is crucial for the evolution of the field. It does not target PSMA. It targets something called NTSR1.
Neurotensin receptor 1.
Right?
Why do we need a new target? I thought PSMA was the magic bullet.
For many patients, it is. But not for everyone. Some patients are PSMA low or even PSMA negative. And then there's the really aggressive form, neuroendocrine prostate cancer, or NEPC.
The shapeshifter cancer.
The very same.
Right.
It drops its prostate identity to evade treatment.
Yeah.
PSMA therapies just don't work for them because the target isn't there.
The house number has been removed.
Exactly. But NTSR1 appears to be enriched in these cases. So this is like a backup plan, a plan B for when the primary target disappears.
A different docking site.
A different docking site. It uses a ligand to attach to that receptor. And bringing it back to our physics lesson, it uses actinium-225 as the payload.
So another alpha emitter.
Yes. Delivering that highly potent confined radiation dose to tumors that might otherwise be completely resistant, it really shows how the field is expanding the address book of targets.
I love that. Expanding the address book so cancer has nowhere left to hide. Now, I want to move into some early trials, phase 1 and phase 1/2, with really novel mechanisms.
This is a very exciting area. We're moving from simply poisoning the cell to manipulating its own survival logic against it.
Let's start with DCC-2812. This one stood out because the mechanism sounds counterintuitive. It seems like you're overloading the system.
That's a very accurate analogy. Cancer cells are already stressed. They're growing too fast. They don't have enough oxygen or nutrients. To survive, they rely on a backup system called the Integrated Stress Response, or ISR.
The ISR is like their emergency regulator. It keeps the engine from blowing up.
Correct. It helps them slow things down and conserve energy. Now, DCC-2812 activates a specific switch in that system called the GCN2 kinase.
Wait, it activates it? Usually we want to block things. Why would we want to help the cancer turn something on?
That is the twist. Instead of blocking it, this drug ramps it up. It activates GCN2, which cranks up the stress response to such an extreme level that the cancer cell simply cannot cope.
So it's like redlining a car engine that's already overheating. You just floor it until it blows a gasket.
That's the perfect image. The cell realizes it's in a state of unrecoverable metabolic crisis and triggers apoptosis, programmed cell death.
It turns the cancer's own survival mechanism into a weapon against it. That's incredibly clever.
It is.
Okay, another one. A phase 1/2 trial for PDS01ADC. This one targets dead zones.
Yes, necrotic regions. This trial combines docetaxel chemo with an immunocytokine called PDS01ADC.
But why target dead tissue? The live cancer is the problem.
Well, when chemo kills cancer cells, it creates these dead zones, this necrosis. PDS01ADC is designed to hunt for things that are normally hidden inside a cell nucleus but get exposed when the cell dies and bursts open.
So the drug only sticks to places where cells have recently died.
Exactly. It uses the destruction caused by the chemo as a landing beacon. And once it lands, it delivers IL-12.
A powerful immune booster. So you kill some cells, which creates a smoke signal. The drug sees the smoke, lands, and tells the immune system, hey, come fight here.
It turns the tumor hot. IL-12 recruits T cells and natural killer cells. The chemo does the initial damage, and the immune system cleans up the survivors.
A smart 1-2 punch. What's the data look like?
It's preliminary, but in 16 mCRPC patients, median progression-free survival was 9.6 months, and 13 out of those 16 patients saw their PSA levels decline.
13 out of 16 is a pretty good hit rate.
It certainly is.
Okay, one more in this section. SHRA1921. This is another 2-ROP2 targeting drug, but next gen.
Yes, another antibody-drug conjugate. The innovation here is the payload. It uses a novel topoisomerase I inhibitor.
What makes it next generation? Is it just stronger?
Stronger, but also more stable in the blood, so it doesn't fall apart before reaching the tumor. And it's better at crossing cell membranes, which creates a stronger bystander effect.
The bystander effect, it kills the cell it hits, plus the guy standing next to it.
Exactly. And that's vital because tumors are heterogeneous. Some cells might have the target, others might not. If the drug can leak out and kill the neighbor, you get a better clearance rate.
The engineers are just constantly refining these tools, making them sharper, more precise.
That's exactly what's happening.
Now, speaking of engineering, let's finish with 2 future tech topics. First up, nanotechnology for bone pain. This is a huge problem.
A major quality of life issue. Bone metastases are incredibly painful.
The source mentions a nanocarrier, Lebanon-Cs at PGSDMB.
Let's just call it the neuroimmunotherapy nanocarrier. This study addresses the crosstalk between bone tumors and nerves. The tumor signals the nerves to amplify pain, and the nerves signal the tumor to grow.
A vicious cycle. The pain isn't just a symptom. The pain signaling actually feeds the cancer.
Essentially, yes. This nanotech is designed to activate only in the high-stress environment of the bone tumor. It delivers 2 things.
Okay, what are they?
First, a sting agonist. Think of it as an alarm bell that turns a cold tumor one the immune system ignores into a hot one that's swarming with immune cells.
And the second thing, it mentioned pyroptosis.
Pyroptosis. It's an explosive form of cell death. The cell membrane ruptures, releasing inflammatory signals. This nanocarrier causes the cancer cells to literally explode.
Cellular fireworks. And the results.
In mouse models, a 94% tumor shrinkage.
94%. That is nearly elimination.
It is. And crucially, it restored bone structure and stopped that pain signaling. It blocked the feedback loop.
If that translates to humans, wow. Okay, finally, the digital side. A new AI tool called AlphaGenome.
This comes back to understanding the basic code of life. AlphaGenome is an AI that can read DNA 1 million letters at a time.
The analogy was that old tools were like looking through a keyhole, but AlphaGenome sees the whole picture.
That's a fair comparison. Previous tools only looked at genes, the parts that make proteins. That's only 1–2% of our genome. The rest is the dark matter.
The so-called junk DNA.
We used to think so, but it turns out that's where the control switches are. In cancer, the problem isn't always a mutation in the gene itself. Sometimes the problem is a switch nearby that's stuck in the on position.
And AlphaGenome can see that.
It can see how the DNA is folded in 3D space, and if that folding accidentally turns on a cancer gene, it helps us find targets we didn't even know existed because we were only looking at the genes, not the switches controlling them.
It's like finding a whole new layer of the blueprint. So what does this all mean? We've covered a lot of ground.
If we connect this to the bigger picture, we are seeing a clear trend. The field is moving away from one-size-fits-all. We're seeing the maturation of theranostics with alpha versus beta. We're seeing a diversification of targets. It's not just PSMA anymore.
It's B7H3, TROP2, NTSR1.
Exactly. And perhaps most excitingly, we're seeing treatments that force cancer cells to self-destruct by overloading their own stress mechanisms.
It feels like we're surrounding the disease from all sides.
We are. And that raises an important question for you to consider. We talked about the PDS-001 ADC trial, which targets the dead zones inside tumors. Think about that. We are reaching a point where we can use the debris of the tumor's own destruction as a beacon to call in further airstrikes.
So the question is,
If we can consistently turn a tumor's death into a weapon against itself, are we finally tipping the balance of the war?
that is a powerful thought to leave us with, turning the enemy's losses into our gains. As always, thank you for guiding us through this, and to our listeners, thank you for tuning in to this deep dive.
It was a pleasure.
Stay strong and fight on.